Good morning, grade 7 students. In this session, we're going to review together the plants chapter in week 24. Green plants, they are the plants, as we said, that contain the green pigment or the green chlorophyll, which is the pigment in green plants that absorb the sunlight. We said that the plants need water, they need mineral salts, they need carbon dioxide, and they need light in order to grow well. Green plants, they need mineral salts. Mineral salts, they are supplied by the water sometimes. Others, sometimes the mineral salts, they are supplied by fertilizers. So fertilizers, they have an important role in supplying the plants or providing the plants with mineral salts. Carbon dioxide, which is CO2. CO2 is found in air. So carbon dioxide, it's tested using the lime water test. If the lime water it becomes turbid, this means that we have carbon dioxide. If the lime water stays clear, this means that there is no carbon dioxide. In this experiment, we observed the different tubes A, B, and C. In tube A, we don't have plant, we have only clay and water. In tube B, we have a plant with root and stem. In tube C, sorry, we have plant with only stem, no roots are available. So here in this experiment, what is the role of the tube A? Tube A, we use it to compare and explain the results. This is called the control tube. So from now on, if I ask for a tube, what is the role of a specific tube? The role of this tube is the control tube. It's used to compare and explain the results. Tube B and C, what is the variable factor between these two tubes? In tube B, we have roots and stem. In tube B, we don't have roots. So the variable factor is the root. So in this experiment, we determined that the structure responsible for the absorption of water in green plants is the root. While if we go closely to the roots, we have the root cap and the root hair. The root cap and the root hair. So here in the root cap and the root hair, we have to go deeply in this experiment. We have in this experiment the root, it's root hair and the root cap, both of them they are dipped in water. The plant one grows normally. However, in plant two, the root hair they are in oil and the root cap in water. The plant withers and dies. So here we can conclude that the plant absorbs the water through the root hairs and not the root cap. So the root hairs, they are the structure responsible for absorption of water. Analysis, if you want to analyze. Analyze and interpret, they are very close to each other, but interpret, we have to add indication. So here, analyze after dipping, always start after. After dipping the root hairs and the root cap in water, the green plant grows. While after dipping the root hairs in oil and the root cap in water, here I mentioned the variable factor, which part of the root is dipped inside the water. So here the green plant withers and dies. If I ask for interpret, what do you have to add? We have to add indication. We have to add this indicates that. If I ask for analyze, this is only the analyze. It's here you stop. Transpiration. What is a transpiration? All of you know that the plant absorbs water through the roots. This water, it travels up through the plant to the stem and then to the leaves. At the level of the leaves, water vapor is lost through the process of transpiration. Before we talk about transpiration, let's talk about stomata. Stomata, they are the small openings on the underside of the leaves. These small openings, they can't be seen by the naked eye. They are seen under the microscope, and these are small openings under the underside of the leaves. So what is the definition of transpiration? Transpiration, it's the loss water vapor to some plant organs. What are these organs? Let's see. Here we have two plants, A and B. The variable factor between these two plants is the leaves. The leaves is the variable factor. We have leaves in plant A while we don't in plant B. We cover both plants with plastic bag and we wait for the results. Water droplets are formed on the inner wall of the plastic bag covering plant A, while no water droplets are formed on the plastic bag covering plant B. So we have release of the water vapor in plant A, which contains the leaves, while no water vapor or no water droplets are formed on the inner wall of the plastic bag covering the plant B. So here, the major difference is the variable factor. It's the leaves. In which a plastic bag the water vapor is formed in the plastic bag covering plant A. What is the part or deduce the part responsible for transpiration? The leaves. Complete the following statement. Through transpiration, green plants they release water vapor from their leaves. You have to keep in mind that the green plant they are called chlorophyllic plants or they are called autotrophs. For example, if I ask for a definition of the chlorophyllic plants, it's not enough to say green plants, chlorophyll plants. It's not enough. You have to say the green plant, they are chlorophyll plants that contain the chlorophyll. You have to mention this. While the heterotroph, they are the non-green plants and they are non-chlorophyll plants. Now talking about photosynthesis. Photosynthesis, as we said before, it occurs only during day in the presence of sunlight. So this process is the food making process. This process where the chlorophyllic plants they produce their organic matter from the inorganic one, from the mineral salts. So the green plants, they produce their organic matter. What is organic matter? They mean about they produce the sugar which is stored in the form of starch from inorganic matter from the water and mineral salts. Food making process of the photosynthesis. In this experiment, we took two plants, A and B, one in light and another in darkness. We boil one leaf from each plant for a few minutes. Boiling the water, it kills the leaf. So boiling water, it kills the leaf. Then we dip each leaf in a tube of warm alcohol to remove its green color while keeping the tubes in boiling water. So warm alcohol it's used to remove the green color of the leaf. Then we soften the leaves by dipping them in water. So boiling the water or boiling water it kills the leaf. Warm alcohol it removes the green color of the leaves. And then we added iodine solution, few drops of iodine solution. Iodine test is done to identify the presence of starch. If we have starch, blue black or dark blue color is obtained. However, if there is no starch, no dark blue color will be obtained. We have brown, yellow, or orange color. This is the test for the iodine test. If we have starch, so plant is undergoing photosynthesis. No starch, no photosynthesis. So as I said, the plant it makes its own food by the photosynthesis. Photo it means light. Synthesis putting together. Putting what? Chlorophyll in the green plant, it traps the energy from the sunlight, it catches the energy from the sunlight. This energy will help to combine, to mix, to put together carbon dioxide and water. It mixes carbon dioxide and water to make the sugar for the plant. This sugar, as I said before, is stored in the form of starch. Here, the reactant in the photosynthesis reaction, they are the carbon dioxide and water. Steps of the photosynthesis. Light, which is trapped by the chlorophyll, it starts the food making process. Then the leaves, they take in carbon dioxide from the air, and the roots, they absorb water from the soil. So we have water and we have carbon dioxide. Then they use the energy from the sunlight, the plants, to make the food, which is a kind of sugar. So it mixes carbon dioxide and water. Then oxygen is considered as a waste product. Oxygen is released from the plant during the photosynthesis process. It's given up by the leaves out to the air. Photosynthesis, it takes place only during the day in the presence of light. The sugar produced is stored in many plants to be stored in the form of starch, where the plant stores it for later use. Here in this experiment, we got a plant, a green leaf. This leaf was covered partially, so a part of the plant, which is this part, was covered by a black cover. So no light will reach this part. So there is no light. This plant was left in sunlight for 2 hours, then it was tested for starch. Tested for starch, meaning that you are testing for the photosynthesis process if it occurs or not. So here we have black, dark blue color or blue black color is obtained for the part that was exposed to the sunlight. However, the part that was covered by the black paper, it shows yellow to brown color, which means that no starch. So if I ask you for interpret, please all of you pay attention to this. Interpret. Interpret, it's not analysis. And interpret, it's not analyze, conclusion, and you write all what you know in this part. Interpret, it's analyze plus indication, and this indication should be specific for the experiment. So here the indication should be specific for the experiment here. So let's start and let's talk about this. After covering this, I'm going to start with the analyze and then I'm going to go to the indication to do this interpret. In order to do the interpret, I have to do analyze part one and then indication part two. And this indication should be specific. And whenever I finish, I'm going to give conclusion. What can you conclude? So start step by step. After covering the plant or the green plant with a black paper and leaving the other parts exposed to sunlight. After 2 hours, we did iodine test. Dark blue color obtained with the part that was exposed to the sunlight. However, a brown yellowish color was obtained for the part that was covered with the black paper. Here, I finished the first part, which is the analyze. Indication. What does this indicate? If we have dark blue color, this indicates that starch is produced in the part that was exposed to the sunlight. However, no starch was obtained or produced in the part that was covered by the black paper. So I'm going to repeat. After covering a part of the plant by a black paper and exposing the other part to sunlight, 2 hours later we did the iodine test. First, the part that was exposed to sunlight, it shows dark blue color. However, the part that was covered by the black paper, it shows a yellow brownish color. Full stop. We have finished the analyze. Indication. This indicates that starch was produced in the part exposed to sunlight. while no starch was produced in the part that was covered by the black paper. Here finished, analyze plus indication. This is what we call it by interpret. Conclusion. Conclusion, it should be in general. So the conclusion, the green plants, they need the sunlight for the process of photosynthesis. So you have to pay attention from now on. From now on, I'm going to be so critical and so accurate in correction. From now on, if I ask for interpret, you did analysis only, you won't take any mark. So this is biology. You have to be accurate. You have to know what is needed in the question. We can't write whenever we want, even though it's correct. We can write whenever we want. Also, a note about the action verbs, I'm going to talk it in the coming slides. Equation, the word equation in the photosynthesis reaction. Carbon dioxide plus water in the presence of light energy, it gives sugar plus oxygen. This is called the word equation. Why it's called the word equation? Because it contains words. Carbon dioxide plus water. I didn't write CO2. If I wrote CO2, this means that it's wrong. Why? Because I asked for word equation. In the presence of light energy, I have to write this over the arrow. Sugar plus oxygen. Moving now to some of the exercises that we had before in the quizzes. Green plants and carbon dioxide. The experiment below is done to study the importance of carbon dioxide for the growth of green plants. Here we have two plants. Plant A grows in a medium rich in CO2. Plant B is poor in CO2. The plant A grows normally. Plant B it withers and dies. So this experiment is done to study the importance of carbon dioxide. So what is the aim of this experiment? Is to study the importance of carbon dioxide for the growth of green plants. What is the pose problem? What is the importance of the carbon dioxide for the growth of green plants? Question mark. Moving for the first part, they said copy and complete the following table of conditions. First of all, the table of condition should include a title and a legend. Title, table showing the conditions and the results. Legend, plus means presence and minus means absence. So here, both plants, they contain water. Sure, the table should be done using a pencil and a ruler for sure. Both of them, they contain mineral salts. Both of them, they contain light. Here we have carbon dioxide. Here we go. Air, it grows normally. And here, normal growth or abnormal growth are the others and dies. So you have to write them for sure. Don't forget the title and the legend. Identify the variable factor. Whenever I said any verb at the rhyme of Wi-Fi, like identify, specify, classify, justify, all of them, verify also, all of them at the rhyme of Wi-Fi, so we have to justify. If we don't justify, you won't get the full mark. What is the variable factor? It's the carbon dioxide. It's present in plant A while it's absent in plant B. What is the objective? To study the importance of carbon dioxide for the growth of the green plants. What is the role of KUH, potassium hydroxide? It's done to absorb carbon dioxide. Interpret the above experiment. So interpret, it's analyze plus indication. So after placing the plant A in a medium rich in carbon dioxide, 15 days later, it grows normally. Why? After placing plant B in the medium B, it's poor in carbon dioxide, the plant withers and dies. This is the analysis. Indication. This indicates that the green plants, they need carbon dioxide or the carbon dioxide is needed for the growth of the green plants. Conclusion, carbon dioxide is important for the growth of the green plants. Here I finish interpret and conclusion. Moving to the Other quiz of the last week. We have observe, document, or figure A. What is the role of the iodine test? Iodine test is done to identify the presence of starch. This is the role of iodine test. Place a geranium plant in sunlight for 24 hours. The plant has green leaves where it's partially covered by a black paper. Immerse the leaf in hot alcohol for 2 minutes, then dip it in iodine solution. The results are shown in the adjacent figure B. Define chlorophyllic plants. Some of you, what did they do? They said chlorophyllic plants, they are the green plants only. This is not a definition. Green plants, they contain the chlorophyll. Here you complete the definition, it's missing. So it said that chlorophyllic plants, they are the green plants that contain the chlorophyll. Part 3. Why we dip the leaves in hot alcohol? To remove the color of the leaves, to remove the green color of the leaves. This is the role of hot alcohol. Part 4. Interpret the obtained results. Interpret the obtained results. If I ask for this, some of you, what did they do? They did analyze, indication, and they give conclusion. I didn't ask for conclusion. This means that you don't know what is interpret. You have to know, guys, that is biology, it's not the science of last year. You have to know that biology is not similar to the science. Biology, it's more accurate. You have to be specific in your answer. If I ask for interpret, you have to do analyze plus indication only. You don't have to add any additional information and you don't have to add a conclusion from your own. This means that you're going to lose the point. And most of you, they did so in the last quiz. I asked for interpret, they did, for example, only analysis. This is analysis, it's not interpret. So the difference between analyze and interpret, that is interpret, it's analyze plus indication. So here, after covering a part of the geranium plant by a black paper and exposing the other part to sunlight, few time or after time, the part that was not exposed to sunlight and covered by black paper, it shows a yellow color with the iodine test. However, the part that was exposed to the sunlight, it shows dark blue color. What does this indicate? This indicates that whenever we have dark blue color with the starch, what does this indicate? This indicates that starch is produced in the part exposed to the sunlight, and no starch was produced in the part that was covered by the black paper. This is the indication here. If I ask for a conclusion, it should be in general. Light is important for the process of photosynthesis. Moving now to give you a quick revision about the graph. Remember when we used to do the graph? The graph, draw the graph that show the variation of the length of the plant as a function of time. Here, they gave you the scale. They gave you the scale. Let's do the graph, the smaller graph here. Where is the x-axis? It's here, it's down. So always the one that's found up in the table should be down in the graph. So here it should be the time in days. And on the y-axis, the one is found down, it should be found up in the graph, length of the plants. Here the length of the plants. In what? You have to put the unit. Length of the plant in centimeter. So here they helped you a lot. They gave you the scale. On the x-axis, every 1 centimeter, it represents 2 days. This is the x-axis, and this is the y-axis. The y-axis is the one, the vertical one. You took this in math. And this is the y-axis. And the one that is horizontal, it's the x-axis. So 1 centimeter, it's 2 days. So here you put 0. the point of intersection. So here, 2, I have to move 4, 2 by 2, 6, then 8, 10, and so on. And the y-axis 1 centimeter, it's 3 centimeter. Okay, so 1 centimeter is 3 centimeters. So here we have to move 3 by 3. So 3, 6, 9, 12, and so on. So now let's move to this graph. This graph is showing the variation of the length of the plant as a function of time. So 2, 4, 6, 8, 10, 12, and the length of plant 3, 6, 9, 14, 18, 21. So I have to obey the scale. If I didn't follow the scale and I use another scale from my own, and they gave you the scale, this graph is going to be cancelled totally. Okay, so you have to pay attention to this. Here is the graph. Let's draw the graph together. First of all, let's put the y-axis and the x-axis. So here we have the point of section is 0. They ask on the x-axis every 1 centimeter it's 2. 2 days. So 2, then we move to 4, then we move to 6. Sure, it should be done using a pencil and a ruler. 8, 10, and this is the 12. What is the title of this axis? The title is Time in Days. And sure, the graph should be done. If you have to do a graph, it should be done on a graph paper. No graph paper, this means that the graph will be cancelled and the mark of this graph also will be cancelled. On the y-axis, every 1 centimeter to present a 3 centimeters. So 3, then I move to 6, and then I move to 9, and then I move to 12. And I didn't put 14 immediately because I'm moving 3 by 3. So 3, 6, 9, 12, and then I have 15, then I have 18, and then I will have 21. Sure, you have to follow the graph paper to be more accurate. I don't have graph paper here, and I'm using my hand, not a ruler. This is the length of plant. Length of plant in centimeter. In centimeter. So this is the first step. Then I prefer to give the title immediately. What is the title of this graph? A graph showing the variation of the length of the plant as a function of time in days. Here you have a graph paper. You choose any of the boxes in the graph paper. And what do? You have to do the scale. Here the scale, you use the graph paper every 1 centimeter. On the x-axis, every 1 centimeter, it's what? It's 2 days. And on the y-axis, every 1 centimeter, it's a 3 centimeter. This is the scale. So here I finished the scale and the title. Here, if you forgot any one of these, I'm going to remove half of the mark. So the scale and the title, they are very important. I prefer to write them before you start with the graph. Let's move now to the graph. At time equal 2 minutes. At time 2 days, sorry. At time 2 days, the length of the plant was 3. This is first. At time equal 2 days, the length was 3. Then let's continue. At time which is equal to 4 days, the length was 6. So 4, remove 4, 6. At time equals 6 days, it was 9 centimeters, the length of the plant. At time which is equal to 8 days, it was 14. Wait a minute, where is 14? It's before 14 is before 15. Here is 14. So I have to add it, and at time 8 it was 14. Then at time 10 it was 18. So this is time 10, it was 18, and at time 12 it was 21, and this is at time 1, 21. I have. It was straight. I can do it using a ruler, but I prefer to connect it using your hand. And this is the graph. Okay, this is the graph. It's done. I draw the graph. First, I gave the title for the axis. I use the scale given. If there is no scale given, you can choose the scale that you want. But sure, you have to mention here whenever you put the scale. Also, don't forget to put the title or the graph. The graph should be done using a pencil for sure and should be done on a graph paper. And I told you before, you have to prepare your graph papers. Here, if I ask you analyze the graph, how can we analyze the graph? It's very easy. At time 2 days, the length of the plant was 3 centimeter. As the time increases from 2 to 12 days, what happens for the length? The length of the plant increases from 3 to 21 centimeter. This is the analysis for this graph.